human genome contains over 20,000 genes, comprised of 3 billion bases of DNA. Unlike our normal alphabet, DNA is written with only 4 letters using a universal pairing rule. A pairs with T, and C pairs with G. Every characteristic and function of the human body depends on the coordinated action of these genes and their products. If genes are to be considered words spelled with these 4 letters, a misspelled gene is the basis for a genetic mutation that can lead to an inherited disease. Technologies such as DNA sequencing have allowed us to learn how these genes are spelled and enabled us to better determine where a mutated or misspelled letter of a gene is located. Pioneering work over the last 20 years using synthetic pieces of DNA called oligonucleotides have formed the foundation of how we can repair or edit a misspelled gene. While these discoveries were paramount in creating the field of gene editing, they served primarily as proof that it is possible to correct a misspelled gene. Only within the last 5 years have researchers discovered a complementary genetic tool that, when combined with oligonucleotides, can effectively correct inherited mutations at a frequency that can make a clinical impact. This new tool emerged from a discovery of a natural system found in bacteria to protect themselves from infection. Some bacteria contain small repeated sequences of DNA called CRISPRs. These sequences are then transcribed into short nucleic acids referred to as guide RNAs. The guide RNAs associate with a protein called Cas9 to cleave the DNA of the infecting agents. This is the CRISPR-Cas9 system. The guide RNAs bind to a complementary target in the invader's genome and construct a scaffold which signals the Cas9 protein to destroy the target. Researchers recognized that this remarkable specificity and efficient cleavage activity could be used to cut human DNA, even in a living cell. These chromosomal breaks are usually rapidly resealed by the cell's own repair mechanism, but the rejoining process is imprecise, leading to the creation of genetic mutations within the cell's genome. These genetic knockouts allow researchers to learn what happens when these genes are disabled. When combined with oligonucleotides, the CRISPR system can be used to repair mutated genes by initiating genetic spell checking. CRISPR-Cas9 cuts the DNA directly adjacent to the mutated base. The single-stranded oligonucleotide then corrects the spelling error. In this way, the mutated base in the gene is removed and the proper base is inserted. The CRISPR oligonucleotide system may be able to target any gene within the human genome, signaling the specific site on the chromosome where a mutated base needs to be corrected. These powerful genetic tools are being used to redesign human cells in order to develop new therapies for inherited diseases, advance the testing of anti-cancer drugs, identify new targets for drug development, and further increase our understanding of the complexity of human disease.